This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas. All right, welcome to the deep dive. Today we're going to be talking about something pretty fascinating and something I think is only going to become more important as time goes on, and that's the connections between fungal pathogens, climate change, and even natural disasters.
Yeah.
And you flagged some really compelling recent scientific research for us, and what we're going to try to do today is really pull out the key insights from these different papers.
Absolutely.
And make sense of it all for you, the learner.
Right.
So to get us started, why don't you tell us a little bit about the research that we're going to be looking at today?
Well, I think the first thing to understand is that, we have to look at how these different forces are all kind of coming together and interacting to kind of impact our health and the environment around us. Mm-hmm. And so, to get started, the first paper we have is from Nature Communications, and the title is Evolutionary Origin and Population Diversity of a Cryptic Hybrid Pathogen. And this paper really zooms in on a specific fungus called Aspergillus latus. Now, it's described as cryptic, and we'll get into that. But it's a very interesting paper that really goes into some depth on this particular fungus.
Yeah, sounds pretty interesting. And then, you also flagged another paper for us, which kind of takes a bit of a broader look at all of this.
Yeah, so kind of taking a step back from just that one particular fungus, we can get a broader perspective from the Lancet Microbe.
Okay.
And they have this great review called Impact of Climate Change and Natural Disasters on Fungal Infections. So this looks at kind of the bigger picture, in terms of how environmental shifts are influencing various fungal pathogens and contributing to disease outbreaks.
All right. And then last but not least, you also sent over a Twitter thread by @Justin46. And while this one is mostly about COVID-19, it does raise some interesting points about co-infections.
Absolutely.
Which, seems pretty relevant for today's topic.
So for you, the learner, the goal of this deep dive is to really, connect these seemingly disparate pieces.
Right.
We want to understand how climate change and natural disasters are shaping the world of fungal infections, including, the emergence and the behavior of specific fungi like Aspergillus latus that we just mentioned, and then really just kind of grasp the broader significance for human well-being.
Right. Okay. So let's dive into the cryptic world of Aspergillus latus first.
All right.
This Nature Communications paper, it gives us a really good detailed look at Aspergillus latus. And one of the things that I found really interesting was that it's an allo-diploid hybrid.
Yeah.
Now, that sounds very technical.
Right.
But in very simple terms, what does that mean for this fungus?
Well, think of it like this. Aspergillus latus has essentially inherited a double set of genetic instructions, a genome that's roughly twice the size and contains twice the number of genes compared to its close relatives.
Wow, that's incredible.
So, the researchers discovered this by looking at its genetic makeup, sequencing the genome.
Right.
And they found that there are 2 copies of most BUSCO genes. Now, these are kind of like essential single copy genes that are usually found in just one version, right?
Okay.
But in A. latus, a surprising 96.68% of these were duplicated.
So almost a complete doubling of these, essential genes.
Yeah, it's pretty remarkable.
So how else did they figure out, or how else did they determine that A. latus was a hybrid?
So they use something called macrosynteny analysis.
Okay.
And so what that does is, imagine you're comparing the arrangement of houses on 2 different streets.
Okay.
Right, to see how similar they are.
Okay.
Macrosynteny analysis does kind of a similar thing with genes on chromosomes. So what they found was that large chunks of the A. latus genome had the same gene order as parts of the genomes of 2 other species, Aspergillus spinolasperis and Aspergillus quadrilineatus.
Wow.
So this strongly suggested that A. latus originated from a fusion of, these 2 or at least closely related species of fungi.
So it's like taking, significant portions of the genetic code from, 2 different fungal species and combining them for fungi.
It's really incredible.
Wow. And was there anything else that that they found, or any other methods that they used to kind of come to that conclusion?
So further evidence came from, phylogenomics, which is like building a family tree
Oh
using
yeah.
genetic information.
Okay.
And so by analyzing over 3,700 of those BSCU genes, they were able to trace the origin of each copy.
Okay.
One copy of each gene appeared to come from A. spinulosporus, while the other came from a close but as yet unknown relative of A. quadrilineatus.
Interesting.
So these duplicated genes that arise from such a hybridization event are called analogs.
Analogs, got it.
Yeah.
So it's not just, a random doubling, but they can actually see, kind of the fingerprints of these two parental lines in
Right.
Elatus. That's some impressive detective work.
Yeah.
And, I'm kind of curious, you have all of these extra genes now, right? So did the fungus just discard the duplicates?
It's interesting that you asked that. So they found that on average, 91.63% of these analogs have been retained in the Elatus genome.
Wow.
So this suggests that, having these extra gene copies has likely provided some sort of evolutionary advantage,
Okay.
and that the genome has become quite stable over time.
Right.
They also found very little evidence of recombination, which is like genetic mixing and matching, between the two sets of parental genes
Right.
across the 22 isolates that they studied that had very complete genomes.
Oh, wow. So it seems like this hybrid fungus isn't, some recent unstable newcomer. What?
No, it's something that has been around for quite some time, and its genetic structure has been established.
It seems so, yeah.
Yeah, that's really fascinating. And speaking of time, I mean, did the paper give any indication of when this hybridization event might have occurred?
They did. So using what are called relaxed molecular clock analyses,
Okay.
which look at the rate of genetic changes over time, they estimate that A. lattice originated somewhere between 13.7 and 13.1 million years ago.
Wow. So it's an ancient
Yeah,
fungus.
it's a very, very deep evolutionary history.
That's incredible.
Yeah, so this hybrid has been around for a very long time.
That's amazing. Okay, so we've got this, really long established hybrid fungus. What else did the researchers uncover about its genes and what they do?
So they looked at, the distribution of core genes,
Okay.
which are present in all the A. lattice samples that they looked at, and
Wow.
accessory genes, which vary between different samples.
And what they found was that core genes are much more common overall.
Okay.
However, the collection of accessory biosynthetic gene clusters, or BGCs, is actually larger than the core BGCs.
And BGCs, those are, they're groups of genes responsible for producing things like antibiotics or other specialized molecules, right?
Exactly. So think of them as like little fungal factories
Right.
that produce various molecules.
Okay.
And what's really intriguing is that the products of many of these BGCs in Elatus are still unknown.
Hmm.
So this suggests that this fungus might have the potential to produce novel compounds, or perhaps some of these gene clusters are just no longer actively producing anything.
Okay.
They also found that the proteins that are produced by core and soft core genes tend to be longer
Okay.
and are more likely to have a known function
Right.
compared to those from shell and cloud genes, which are more variable.
Okay.
And surprisingly, the softcore BGCs actually contain fewer genes on average than the other types.
Oh, wow. Interesting. So the consistent set of genes, in Elatus is pretty well understood, but the more variable genes, particularly in those BGCs, are kind of a mystery box in terms of what they can do.
That's a great way to put it.
Yeah, and that just sounds like, a really fertile area for future research.
Oh, absolutely.
Okay, so what about how these genes are actually used? Is there a preference for, the genes from one parental genome over the other?
Well, that's a really insightful question. So they investigated gene expression levels at both 30 degrees Celsius and 37 degrees Celsius,
Okay.
which are relevant temperatures, for its growth in the environment and potentially in association with, mammals.
Right.
What they discovered was that the transcript abundances, which basically tells us how much of each gene's blueprint is being used to create proteins, like how often a recipe is being followed in a kitchen,
Right.
were almost equal from both the A. spinulosporus-like
Right.
and the A. quadrilineatus-like parts of the genome at both temperatures.
Gotcha.
They also observed that codon usage bias, which can influence how efficiently a gene's message is translated into a protein,
Okay.
was similarly optimized in both subgenomes.
So it's a pretty balanced, operation with both parental contributions actively involved. in the fungus's life at those temperatures.
Yeah, it's really remarkable.
Yeah. Did they see any shifts in gene usage when they changed the temperature?
Yes, so they did a differential expression analysis,
Okay.
and what they found was that there was a similar number of genes that were significantly more or less active in each of the parental genomes
Okay.
when they shifted between 30 degrees and 37 degrees Celsius.
Okay.
Interestingly, many of the analogs that showed differential expression
Right.
displayed what are called conserved responses, meaning both copies of the duplicated gene reacted in the same way
Right.
to the temperature change.
Okay.
For example, they saw an increase in activity of genes involved in the glutathione metabolic process,
Oh.
which is known to help organisms cope with heat stress.
Yeah, that makes sense, kind of a coordinated defense against a changing environment.
Exactly.
But were there also instances where, the analogs behave differently?
Absolutely.
Where one copy of the gene responded to the temperature shift and the other one didn't?
Right. So they observed subgenome-specific responses as well.
Okay.
In some analog tears, one copy from one parent would become more active, while the copy from the other parent might not change or even become less active.
Interesting.
So this indicates that both parental genomes are contributing to the overall functioning of the fungus,
Right.
but they also have their own distinct ways of responding to environmental cues.
Right. So it's kind of like this, this hybrid nature gives it a broader range of tools to handle different, different conditions.
Absolutely.
So it's got, shared and specialized responses, from its duplicated genes.
Exactly.
Yeah, that's a pretty cool, setup.
It's a very, advantageous setup for sure.
Yeah. Okay, so now let's circle back to why it's called a cryptic pathogen.
Right.
What makes it cryptic and what are the implications of that?
Well, so the study really emphasizes that this detailed genetic characterization that they've done is really vital for developing accurate diagnostic tests.
Okay.
And the reason for that is because A. latus can be quite difficult to distinguish from other Aspergillus species just based on its physical appearance alone.
Okay.
So these specific genetic markers can really help us improve our ability to track its spread and understand its actual role in causing disease.
So we need to be able to really identify it to know how prevalent it is and what kind of health risks it might pose.
Absolutely.
Okay, and this leads us to the interesting connection with COVID-19 that came up in that Twitter thread.
Right.
Yeah.
So the researchers noted that 6 clinical isolates, basically samples taken from patients, that were initially misidentified as A-latus, were actually found to have come from individuals who were also infected with COVID-19.
Oh, wow.
So while there's already some research on Aspergillus fumigatus as a co-infecting agent in COVID-19 patients,
Right.
the discovery of these A-latus co-infections adds to the growing body of evidence that other Aspergillus species can also establish themselves in patients who are battling SARS-CoV-2.
Yeah. That's a really interesting connection.
Yeah.
And it really, if we're seeing these fungal co-infections, in the context of viral illnesses, it really underscores the importance of the kind of accurate identification that this study makes possible.
Absolutely.
Yeah. Okay, so that was a really fascinating deep dive into Aspergillus latus. Now let's shift gears a little bit, and look at the broader impact of climate change on the fungal world, drawing on some of the insights from the Lancet Microbe Review.
Yeah, so this review really highlights how the long-term shifts in temperature and weather patterns that are caused by climate change are having a far-reaching impact on pathogens, their hosts, and where they can survive.
Okay.
And as the authors point out, it's often the populations who are most vulnerable to infectious diseases who are going to bear the brunt of these climate-driven changes.
Yeah, that's a sobering thought.
Absolutely.
And, with regards to fungi specifically, the review really emphasizes that many pathogenic fungi, they're not just surviving these increasing global temperatures, they're actually thriving and adapting to the heat and potentially becoming more dangerous.
Exactly. And a key example that they discuss is Candida auris, this relatively recent fungal pathogen that has demonstrated significant tolerance to higher temperatures, including those within the human body. So the review also points out how climate change has likely contributed to the expansion of endemic fungal diseases like coccidioidomycosis and histoplasmosis into new geographical areas,
Okay.
as well as affecting fungi that impact animals and plants.
Okay.
And we can't overlook the fungal pathogens that attack plants, which are also evolving in response to climate change and posing an increasing threat to our food supply.
Right, right. So it's not just about, the direct threats to human health, but it's also, the consequences for agriculture could have,
Right.
Significant indirect impacts on us as well.
Absolutely.
Yeah. So how exactly are these fungi managing to adapt to all of this increasing heat?
Right.
What are some of their strategies?
Well, the review details several, pretty remarkable mechanisms.
Okay.
One important one is the increased production of heat shock proteins, or HSPs,
Okay.
which help to protect other proteins from damage at high temperatures.
Right.
They also mention the activation of specific signaling pathways like HSS, HOG, and calcineurin signaling, which help the fungus cope with various stresses. Additionally, they note increased expression of amino acid permeases,
Okay.
which are involved in taking up nutrients,
All right.
and the enhanced production of melanin, that dark pigment that we often see in fungi.
Right.
Melanin actually acts like a shield, providing protection not only against heat, but also against other environmental stresses like changes in pH, heavy metals, and even radiation.
So it's kind of like a fungal, Swiss Army knife,
Right.
offering defense against multiple threats.
That's a great way to put it.
Are there any other kind of adaptations that they're using?
So they're also modifying their cell walls, making them more robust and less permeable.
Okay.
Some fungi are even changing their ploidy, essentially increasing the number of copies of their entire genome.
Wow, that's incredible.
Increased mutation rates,
Yeah.
possibly driven by the activation of mobile genetic elements,
Right.
can also accelerate the pace of adaptation. And they're observing enhanced morphogenesis, which just means changes in their physical form, as well as increased formation of biofilms.
Right.
Those sticky communities of microbes that can be very resistant to both antifungals and the host's immune defenses.
Right.
Finally, they can ramp up the production of enzymes that detoxify harmful substances and metabolites that help stabilize their proteins.
So they've got, that's an incredible array of adaptive strategies.
It's quite impressive, actually.
Yeah, it really is. And the review suggests that these adaptations, not only help them survive the heat, but they can actually make them more harmful to us.
Precisely, yeah. So these mechanisms can lead to increased virulence, meaning they're better at causing disease,
Right.
and an enhanced ability to evade our immune systems,
Right.
making it harder for our bodies to fight them off.
Yeah, that's not good.
No, not at all.
So the review also touches on how climate change can influence other factors that affect fungal ecosystems and our own susceptibility to infections, things like pollution, microplastics, and even changes in UV exposure.
Yeah, so they highlight that pollution in water bodies can actually create more favorable conditions for fungal growth and increase fungal diversity, including pathogenic species.
Okay.
And the reason for that is that some pollutants can actually provide a food source, or they can alter the environment in ways that benefit fungi. This is actually a concern even in industrialized nations.
Right.
The role of microplastics is also really interesting. So while some fungi can actually break down plastics, the widespread presence of microplastics in water could inadvertently provide a surface for fungal growth.
Right.
And potentially even contribute to the development of antifungal resistance.
Oh, wow.
Yeah, it's kind of a double-edged sword.
Yeah.
Increased UV exposure, which can be a consequence of, a thinning ozone layer due to climate change,
Right.
can negatively impact human immunity, making us more vulnerable to infections in general.
Right. Okay.
And they even mention a slight decrease in average human body temperature,
Oh, wow.
which could theoretically affect our natural resistance to certain fungi.
That's really interesting about a human body temperature. And, the review also draws a connection between climate change, increased fungicide use in agriculture, and the rise of antifungal resistance in human pathogens.
This is a really critical point.
Right.
So as climate change is driving the evolution of fungal plant pathogens, there's an increased reliance on fungicides in agriculture to protect crops.
Right.
Unfortunately, many of these agricultural fungicides belong to the same chemical classes as the antifungals we use in human medicine,
Oh, wow.
particularly the azole class.
Right.
So this widespread environmental exposure can drive the development of resistance in human pathogens like Aspergillus fumigatus
Right.
and Fusarium sipi.
Right.
And even more worryingly, some of the newer fungicides being developed for agriculture share their mechanisms of action with novel antifungal drugs that are currently in development for human use.
Oh, wow.
So this could potentially lead to resistance to these new drugs even before they become widely available.
That's a pretty scary prospect.
It's deeply concerning.
Yeah, potentially losing these future antifungal treatments, before we even have a chance to use them.
Yeah,
Because of resistance that's developing in agricultural settings.
exactly.
Yeah. So it seems like climate change is really driving, shifts in fungal infection patterns in a few different ways, right? We have the emergence of new threats.
Right.
We have the spread of existing ones, and then we have this increasing difficulty in treating them due to resistance.
Exactly.
Yeah, it's a pretty complicated situation.
It's definitely multifactorial.
Yeah. So the review emphasizes that warming temperatures are a key factor in the emergence of new human fungal pathogens.
Yeah.
With the ability to tolerate higher temperatures being, really critical.
Yeah.
Right. They provide examples like Candida orthopsilosis, Fusarium oxysporum, Cryptococcus deuteragadii, and several species of Bipolaris and cladosporin.
Right.
And as we discussed earlier, Candida auris is, a prime example
Yeah,
of a
exactly.
pathogen that's hypothesized to have emerged, at least in part, due to these rising temperatures.
Right.
And it's not just, entirely new species,
Right.
but the geographical ranges of established and endemic fungal diseases are also, on the move.
Absolutely, yeah. So they discuss the expansion of endemic mycoses like coccidioidomycosis, histoplasmosis, and blastomycosis into regions where they haven't historically been prevalent. Even the way that fungal spores are dispersed through the air can be affected by warmer temperatures and increased atmospheric turbulence,
Right.
influencing the distribution of fungal pathogens.
Okay.
And they also note that several skin and eye fungal infections, which thrive in warm and moist environments, are also likely to be influenced by climate change.
Yeah. So climate change is really, acting as this powerful force, reshaping the entire landscape of fungal diseases.
It really is.
Yeah. Okay, so let's move on to the third piece of our puzzle today, which is natural disasters.
Right.
The Lancet Microbe Review also explores how, these events, which are becoming more frequent and intense due to climate change, can trigger outbreaks of fungal diseases.
Yeah, and this is a really crucial connection to understand, because natural disasters often create this perfect combination of factors that can dramatically increase both our exposure to fungal pathogens and our susceptibility to infection.
Oh, okay.
So it's kind of a double whammy.
Right. So the review outlines several different ways this can happen.
Yeah.
And I think one of the most direct is through traumatic injuries that provide an easy entry point for fungi into the body.
Exactly. So cuts, scrapes, any kind of break in the skin's protective barrier can allow fungi from the environment to gain access. And the contamination of wounds with organic matter, which is common after disasters, can lead to serious mold infections in the skin and soft tissues.
Yeah, that makes sense. And beyond those physical injuries, natural disasters, they also take a toll on our immune systems.
Absolutely. So the immense psychological stress and the physical trauma associated with disasters
Right.
can weaken our immune defenses,
Right.
making us much more vulnerable to opportunistic fungal infections.
Right. The ones that wouldn't normally
Exactly.
cause a problem in a healthy person.
Right.
Right.
And then there's the issue of fungal spore dispersal.
Okay.
Events like wildfires, volcanic eruptions, and strong winds can release vast quantities of fungal spores into the air,
Right.
potentially leading to widespread exposure and respiratory infections, even in populations far removed from the immediate disaster zone.
And then you have the conditions in the aftermath of, floods and heavy rainfall
Right.
that can also be ideal for fungal growth.
Yes, the increased humidity and the water damage to buildings create a perfect breeding ground for molds.
Right.
And this can significantly elevate the risk of respiratory problems and chronic diseases due to that prolonged mold exposure.
Okay.
The review even presents a compelling hypothesis about tsunamis,
Oh, wow.
potentially introducing pathogenic waterborne microbes to land, which may have been the case with Cryptococcus gattii in the Pacific Northwest following a major earthquake in Alaska back in 1964.
So a major, geological event having these, long-term biological consequences in a distant location. That's pretty fascinating and also, a bit unsettling.
Yeah, definitely.
Okay, so the review also provides, some specific examples of fungal outbreaks that have followed natural disasters.
They do. So they mention cases of severe soft tissue infections caused by the fungus Rhizopus aureus after a volcanic eruption in Colombia in 1985. The devastating Indian Ocean tsunami in 2004 led to a number of cases of invasive mold infections linked to fungi like Apophis elegans and Aspergillus. More recently, we saw an increase in invasive mold infections after Hurricane Harvey in Houston in 2017.
Right.
And then there's the tragic example of pulmonary hemorrhages in infants that were linked to the mold Stachybotrys chartarum after flooding in 1994.
Wow.
And even the long-term health consequences of hurricanes like Katrina and Sandy include an increased risk of respiratory symptoms and chronic diseases from mold exposure in damaged homes.
So it really illustrates how these large-scale environmental catastrophes can have these very specific and sometimes unexpected impacts on fungal infections.
Absolutely.
Yeah, and the review also touches on the effects of wildfires on soil ecosystems.
So wildfires can drastically alter the diversity and the function of microbial communities in the soil.
Right.
And this includes fungal populations. And there's actually this emerging field of pyroaerobiology, which is specifically investigating how viable microbial life can be aerosolized and transported by smoke from wildfires, potentially influencing the distribution and the spread of fungal species.
So it sounds like, fungal outbreaks following natural disasters are a pretty significant public health concern.
They are.
And perhaps one that doesn't get the attention that it deserves.
Yeah. And the review actually explicitly suggests that these outbreaks are likely underreported. This could be due to a number of things. The overwhelming challenges in disaster-stricken areas where immediate survival needs take precedence, a lack of readily available diagnostic tools and trained healthcare professionals to identify fungal infections in these chaotic settings, and unfortunately limited research funding directed towards this specific intersection of disasters and fungal disease.
Right. So we may only be seeing the tip of the iceberg when it comes to the true burden of fungal diseases following natural disasters.
It seems very likely.
Yeah. Okay, well, this has been a really in-depth and fascinating exploration of how all these factors are interconnected.
Yeah, it's really it's an incredibly complex and important topic.
Yeah, it really is. So let's wrap up with some key takeaways for you, the learner.
Absolutely. So, we've seen how Aspergillus latus is this really compelling example of a cryptic hybrid pathogen with a long evolutionary history and even the potential to co-infect COVID-19 patients.
Right.
This highlights the hidden diversity within this Aspergillus genus and the possibility of, a lot of underdiagnosed infections.
Right.
So understanding its genetics is really key to better diagnostics.
Right. We've also learned that climate change is this, really powerful driver in the adaptation, emergence, and spread of a wide variety of fungal pathogens. Yeah. They're becoming more tolerant to heat, potentially more virulent, and expanding their geographical ranges, posing new threats to both human health and agriculture.
Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other podcasts, responding to the content, and checking out our related articles at helioxpodcast.substack.com.